The development of the Morse code in the mid-1800s revolutionized the way people communicated. Using a coded series of dots and dashes to represent letters of the alphabet, information that once took days or weeks to deliver could be transmitted in a matter of minutes. People who are blind or deaf also make use of codes. Telephone decoding machines called TTYs are used by people who are deaf and hard of hearing to send and receive typed messages along phone lines. The Braille alphabet helps people who are blind communicate by making the connection with the spoken word. One of the basic communication codes is the characters or symbols that comprise language. A computer modem uses a code which turns the letters on the screen into electronic signals that can travel down the phone line to another modem, where the code is translated into recognizable words. And, although you can't see it, similar coding and decoding is going on in your body all the time. Without this information transfer, living systems could not function, reproduce, or survive on this planet.
Program, we will examine how the cells produce proteins using DNA as a blueprint for their construction, just like a builder uses a blueprint to build a house. The blueprint provides a unique set of instructions that specify the shape and function of each room. If any part of the blueprint is altered or damaged, the design of the room will be changed or misinterpreted. Similarly, our genes provide a unique set of instructions that code for the synthesis of a specific protein. If any part of the gene is altered or damaged, another protein with a function different from the original desired protein will be synthesized. We'll also look at amino acids, the building blocks of all proteins. And we'll learn how what we eat affects the body's ability to translate the DNA into the proteins that are essential for life.
A short review of DNA structure and function will help you to understand protein synthesis and the way that amino acids are involved in the formation of a protein. DNA is arranged in a spiral known as a double helix. If you're to unravel it, however, DNA would look more like a ladder. The uprights or sides of the ladder consist of a deoxyribose sugar and a phosphate group. The rungs are pairs of nitrogen bases joined end to end. There are 4 different nitrogen bases: adenine, guanine, thymine, and cytosine. DNA is actually built from units called nucleotides, which consist of a nitrogen base, a sugar, and a phosphate group. DNA can make identical copies of itself in a process called replication. It can also produce a single complementary strand known as messenger RNA. This latter process is called transcription. The double-stranded DNA unravels. One strand of the DNA is used as a template for the synthesis of a complementary strand of RNA. Temporary base pairing occurs between the DNA template and RNA nucleotides. When the termination site on the DNA template is reached, the single strand of mRNA is released, and the DNA molecule returns to its coiled shape again.
You know, it's amazing how something as simple as a sequence of these bases can spell out the entire genetic code.
You're right, Vanessa. It is amazing.
And I thought it was interesting that DNA is used as a template for RNA.
Well, RNA isn't exactly identical. See here. RNA has a different sugar and also a different base than DNA.
Right. The differences between them are DNA has a deoxyribose sugar and a base of thymine. RNA has a ribose sugar and a base of uracil. You know, I still don't understand how these two are used in the production of proteins.
I think the sequence of bases forms some kind of code.
I think I may have something here that will help you understand. Code is really essential for cells so that they can produce the protein that they need to function normally. Two examples come to mind. These cells of the digestive tract, which need to make enzymes, and the cells of an endocrine gland, which need to make hormones such as ADH, or insulin, oxytocin. Now, the code is a triplet code, see? These groups of 3 nitrogen bases. And I'll show you on this paper representation of an mRNA strand as well. AUG, UGU. These are groups of 3 nitrogen bases, and each group of 3 bases is called a codon. And all the codons except for 4 code for a specific amino acid.
So proteins are made up of amino acid chains.
Right. Amino acids are small organic compounds that bond together to form a protein. There are 20 amino acids, and we need all 20 of them to form all the proteins that are required to keep our body functioning normally.
Do our cells make all 20 amino acids?
All but 8.
The 8 essential amino acids.
Right. Your body can produce 12, but the other 8, the essential amino acids, must come from the food that you eat.
So what do amino acids look like?
Looks like this. All amino acids have the same basic structure. They have an amino group with 2 hydrogens and an acidic carboxyl group, which is a carbon double bonded to an oxygen and a hydroxyl group down here. Now, the differences come from the atomic structure of the R group. In the simplest amino acid, the R group is a hydrogen atom. That's the amino acid glycine. A peptide bond is formed when the functional groups of 2 amino acids, the amino group and the acidic carboxyl group, react in a process called dehydration synthesis. These atoms are removed, a peptide bond is formed, which we'll represent by this red line.
So because it's called dehydration synthesis, the water is lost.
Good for you. 2 hydrogens and an oxygen is a water molecule. Why don't you try some dehydration synthesis? I'll get some more interesting R groups. This is the R group for the amino acid cysteine. This is the R group for the amino acid tyrosine. Benaz, if you can take out that R, we'll replace the R with this. Great. This is the R group for phenylalanine. Thanks, Cam. And this is the R group for the amino acid called glutamic acid. Thanks. Okay, why don't you try some dehydration synthesis?
Okay, well, the amino group in this amino acid and the carboxyl group in this amino acid will react to form a peptide bond which removes the water.
And the same is true for these two. The amino group and the carboxyl group react. Water's lost. And a peptide bond is formed.
Great. And don't forget, this carboxyl group and that amino group are still both open to further reaction.
So how many amino acids can make up a protein?
Well, anywhere from under 10 to about 1,000. But what's interesting is that these 4 amino acids in this sequence form the first 4 amino acids of a very short protein molecule, only 9 amino acids. It's called ADH. And because the amino acids are joined together by peptide bonds or linkages, proteins are sometimes called polypeptide chains.
Can we go back to the code for a minute?
Yes, is there something confusing you about it?
How do we know it's a triplet code? I mean, why couldn't it be a doublet code or a quadruplet code?
That's a very interesting question, Cam. Let's take a look at the numbers. Let's say, for example, it's a singlet code. That means 1 nitrogen base would code for 1 amino acid. Well, then we could only have 4 amino acids because there's only 4 bases.
And if it was a doublet code, then that would be 4 squared, so that's 16 different possible combinations coding for 16 different amino acids. So, for example, here we have AU, GU, GU, CA, and so on.
Those are the 16 bases. What if it were a triplet code?
Well, if it were a triplet code, that would be 4 cubed, or 64.
I have a list for that, too. These would be the triplet codes on the DNA strand, but you can see that there's a lot more. Okay, one step further. What if it were a quadruplet code?
So that's 4 to the 4th power. So that's 256 possible combinations. So, did you make a table for that as well?
No, I didn't. I stopped where the scientific research decoded the code, at the triplet code.
So, a doublet would form 16 combinations, which is too few because we need 20 combinations of amino acids.
But the quadruplet code provides too many possible combinations. So who decided on the triplet code?
Well, scientists in the mid-1960s discovered that there were 64 codons that functioned in producing proteins. As it happens, some amino acids are coded for by more than one codon. Arginine, for example, has 6 different codons: CGA, CGU, CGC, CGG, AGA, and AGG. And you can see some of these other amino acids have multiple codons as well. This provides for some redundancy in the genetic code. More than one codon can code for a specific amino acid, and the variation in the possible combinations of these 64 codons allows for all 20 amino acids to be coded for. The building blocks of proteins are molecules called amino acids, of which there are 20 different types. It can be thought of as a 20-letter alphabet, used in specific combinations to form words, or in this case, proteins. Each set of 3 bases in the mRNA specifies an amino acid, represented here with a 3-letter abbreviation. To initiate and terminate protein synthesis, there are 4 codons. AUG is the start codon, while UAA, UAG, and UGA are the stop codons. Now we know how the triplet code works. You ready to see how protein synthesis occurs?
Sure. I know what happens after transcription, but what happens next?
The next process is called translation, and it occurs in the cytoplasm. The newly formed mRNA strand, the messenger RNA, that's just been formed from transcription, migrates from the nucleus to the cytoplasm. But it's not quite ready yet.
What do you mean it's not ready?
There are 2 forms of messenger RNA, a premature form and a fully functional form. On the DNA molecule, there are coding regions called exons and some non-information regions called introns. Now, during transcription, those introns are transcribed onto the messenger RNA. So before it can be fully functional, those non-information regions need to be edited out. Once the editing processes are complete, then the messenger RNA can be fully functional and be used as a guide in translation.
Kind of like translating a message from one language to the other.
Exactly like that, Cam. But in this case, what are you translating?
The sequence of bases into the sequence of amino acids.
Which then determines
the type of protein produced.
Exactly, Banaz.
But where in the cytoplasm does this take place?
Aren't the ribosomes the site of protein synthesis?
They are, Banaz. So the fully functional messenger RNA migrates from the nucleus to the ribosome. Then what?
Well, somehow the triplet code has to pick up the correct amino acid. But how does that happen?
Okay, to understand how that works, we need to look at the other 2 types of RNA. Now, one type is called transfer RNA, or tRNA. Any guesses what its role is from its name?
Transfers or transports the amino acid in the cytoplasm to the messenger RNA on the ribosome?
Good thinking, Cam. Each transfer RNA molecule is specifically designed to carry only one of the 20 amino acids. Enzymes are responsible for linking the amino acids to the correct transfer RNA molecule. A typical tRNA molecule looks like this. It's got a 3-dimensional shape. It's often described as a clover leaf because of these base pairing regions.
So where does the amino acid attach itself?
It attaches here, Banaz, on the acceptor end. There's a sequence of bases here, ACC. That's common to all tRNA molecules. And the amino acid binds there.
So does the tRNA actually attach itself to the mRNA on the ribosome?
It does. This loop down here on the tRNA, it's a special site called the anticodon. It has a specific complement on the mRNA strand. And when the anticodon on the tRNA recognizes the codon on the messenger RNA, temporary hydrogen bonds are formed. Now, here's the attachment of the amino acid on the tRNA molecule on this acceptor end. The transfer RNA molecule's role in translation is to match the codon to the appropriate amino acid.
So then that must mean that if the mRNA codon is UGU, then the corresponding bonding anticodon is ACA?
That's right, Benez. So what would be the anticodon for UAC?
Wouldn't it be AUG?
Right.
Where does the amino acid come from?
When the foods you eat are digested, the amino acids that result are absorbed from your digestive tract into your bloodstream, and then they're transported to all your body cells. Or amino acids can also be manufactured in your body cells from the conversion of fats or carbohydrates, nucleic acids, even other amino acids.
So then the amino acids are already in the cytoplasm, ready to bond?
Yes, they are. Okay, now let's look at the third type of RNA, which is ribosomal RNA, or rRNA.
It must control what's happening at the ribosome.
Yes, it does, Cam. This is a ribosomal complex, and it's made of ribosomal RNA and proteins. There are two subunits to this ribosome, a large subunit and a smaller subunit. Now, as it moves along from the initiator end, along the messenger strand, another ribosome can move in at the initiator end, so that several ribosomes can be translating at once. And when that happens, it's called a polysome. Let's just use this one ribosome, and let's build a protein molecule.
Okay, well, we already know that the codon UGU matches with the codon ACA, and the codon of the mRNA is UAC, so the anticodon is AUG. So that's this one. So we'll put it right here.
What's this bond called up here between the amino acids?
Peptide bond?
And what's the process?
Dehydration synthesis.
Good memory. Okay, then the ribosome moves along, and the transfer RNA detaches itself from the amino acid and goes back just into the cytoplasm.
Is that where it picks up the next amino acid?
It picks up another one of its own specific amino acids.
So for this one, UUC, the anticodon would be AAG, which is right there.
Right. So we have our peptide bond. And the ribosome moves along. And we lose our transfer RNA into the cytoplasm.
So GAA would be CUU. Peptide bond. Ribosome moves along.
And we lose our transfer RNA.
AAU is UUA. Ribosome moves along. Uracil, guanine, uracil is adenine, cytosine, adenine. This is fairly easy once you get going.
Makes sense.
It goes a little quicker in the cell.
CCC, GGG. CGU, GCA. GGU, last one, CCA, and on to the terminator, UAA.
Great. Now you've just made a 9 amino acid protein. That's an actual protein. This is the sequence of amino acids in the protein ADH, or antidiuretic hormone. The only thing that isn't shown here is that there would be a disulfide bond between this cysteine and this cysteine. Good work. You've just done a protein synthesis.
Can you believe? We just made a molecule. I think she's here, Cam.
I'm just going to finish up the table while you let her in.
Okay. Hi, glad you can make it.
Hi, Vanessa. Your directions were great. What is this?
Well, it's our homework assignment.
Oh, it smells great. I don't recall assigning food.
Well, that's because we kind of assigned this to ourselves.
I like the look, Cam.
Thank you. Do you remember when you were telling us how important amino acids are to our bodies?
Yes.
Well, we decided to serve up a protein-rich meal so we can all stay healthy. Come join us at the table.
Oh, this is great.
We did this just to prove that we were really paying attention in your class. So, if you'll have a seat, we'll show you what we've prepared.
By all means.
Your napkin.
Oh, my.
Thank you.
Here you'll find several tasty dishes featuring lentils, grains, and corn.
Now, we know that individually, one of these would not give you your complete range of amino acids. So that's why we have such a wide range of legumes, grains, and corn. This way you'll get all the protein and the 8 essential amino acids.
Well, you've obviously done your homework. I know I will receive the 8 essential amino acids from this meal. What about the other 12 amino acids? Where will they come from?
Non-essential amino acids can be obtained from our diet, or if our diet is deficient in one or more of these amino acids, then they can be produced from molecules derived from carbohydrates, fats, and proteins. Now, all 20 amino acids are then used to make proteins which are required for cell metabolism, tissue development, repair, and growth.
One more question. This smells so good. Could we eat?
Absolutely.
Sure.
Why are proteins and amino acids so important to someone who's strength training?
Strength training athletes need to consume extra protein so that they're able to build the muscle mass and build the strength that they want to build. But what's interesting is the most important factor for building strength and building muscle mass is exercise. It's not diet.
What is the daily requirement of protein for someone who is strength training?
Well, for a strength athlete, they need about twice the protein requirement than, say, what a sedentary person would require. But what's surprising is that already Canadians are consuming, on average, twice the protein that they need. So in other words, strength athletes just need to eat the same amounts that normal people would be eating.
Do some athletes require amino acid supplements?
Really, there's no athlete that requires amino acid supplements. A lot of athletes might take them, but again, they're not necessary. Simply eating food, adequate amounts of food. For example, a strength athlete, if they were to have even, say, 3 ounces of meat, this is uncooked, or 3 ounces, here's a chicken breast, if they had 2 of these a day with a couple glasses of milk and then some grains such as pasta or rice, that's ample protein that they need without taking supplements. A lot of athletes in the gym might boast about the success that they've had with their training because they're taking these supplements. But what the athlete doesn't realize, if they didn't take those supplements, they'd still have the great gains that they're making in their fitness. So a lot of it is testimonials, athletes just saying, it works for me, you should take it too, it'll work for you.
What about the negatives?
Negatives, I guess my biggest concern is the athlete would focus so much on protein, and yet the main fuel that they need for training is carbohydrate. So they might not eat enough carbohydrates, but instead eat huge amounts of protein. And that can reflect problems with, say, dehydration from too much protein or calcium excretion. And that may lead to maybe even things like osteoporosis. But generally, it's just extra calories, and the athlete might end up just storing it as fat.
In the nucleus, a DNA segment or single gene specifying one protein uncoils and a single-stranded mRNA is transcribed from the DNA template. mRNA moves from the nucleus to the cytoplasm. When mRNA attaches to the ribosome, translation begins. The mRNA can attach to several ribosomes, but to simplify the process, we will only refer to one. Each amino acid attaches to its proper tRNA with the help of a specific enzyme and energy in the form of ATP. 2 tRNAs fit into the ribosome complex at one time. This allows the 2 amino acids to chemically bond to one another. Dehydration synthesis occurs, and a peptide bond forms. A succession of incoming tRNAs carrying their amino acids bond with a specific codon of the mRNA on the ribosome. As the ribosome moves along the mRNA, a new amino acid is added to the polypeptide chain by dehydration synthesis. The released tRNAs recirculate in the cytoplasm, ready to be matched with another amino acid. The polypeptide chain is released when a stop codon is read in the mRNA. A human has many different kinds of proteins, each with a specific structure and function. The function of a protein depends on the sequence of amino acids making up its polypeptide chain. As you can see, the fibrous proteins play an important role in the structural framework and muscular contractions. Globular proteins include enzymes and transport proteins. Enzymes catalyze important reactions in your body, while hemoglobin, a transport protein, carries a fresh supply of oxygen to our cells. Globular proteins also specialize in regulating pH and metabolic rate. They play a crucial role in maintaining homeostasis. Finally, certain globular proteins defend us against foreign substances. For instance, antibodies are produced to inactivate harmful substances that enter our bodies.
Whether we're talking Morse code, modem, blueprints for a house, or protein synthesis, we're talking about the same thing: the coding and decoding of information to make it accessible and useful. You have learned how our genetic blueprint is transcribed in the nucleus into messenger RNA and translated in the cytoplasm. The sequence of bases on the DNA molecule is crucial to this process of translation. Each messenger RNA codon codes for a specific amino acid, which then forms a peptide bond with each successive amino acid as the polypeptide chain is synthesized. The outcome is a protein, and proteins are necessary for our well-being. Although there are countless number of proteins synthesized in our body, they are all made from just 20 amino acids. 12 of these amino acids can be produced in our body. The other 8, called essential amino acids, have to be taken in with the food we eat. All 20 amino acids will form the backbone of the proteins which are necessary for the maintenance of cell life.